Okay.
And your last name is Level?
Lavelle. Yeah.
Lavelle. Okay, good. Good.
Yeah.
That's good to get fixed. Okay. Okay, so I'm just going to quickly introduce you just by name, and then we'll go into the questions, and we'll roll from there.
Sure.
And if any time you want to stop, just throw your hands up and just say cut, whatever you want to say.
Okay.
Okay, so I always have to take a deep breath. I do get a little nervous about these sometimes. Okay, I'm going to bring it back to me. Okay. It's my pleasure to be speaking today with Julian Lavelle. Dr. Lavelle is a scientist at Novartis, and so my first question to you, Dr. Lavelle, is what is your position at Novartis?
I'm a medicinal chemist in the oncology group, actually, at the moment, although I've actually moved between several different disease areas in my career. My title, I guess you could say, is a senior researcher or group leader.
Excellent. So how did you come to hold this position at Novartis?
Obviously, I followed a normal path in graduate school, did my PhD. I then did a postdoc, actually over in the States. My PhD was in England. And then after that, I got my first job at a biotech company for a couple of years. And then I moved into the big pharma companies, originally with Aventis, and then 10 years ago I joined Novartis. And I've been here up in Boston now. I'm based in Boston for the last 9.5 years.
Okay. So what are the day-to-day activities that define your position, typically, if you have a typical day?
So, I mean, as a medicinal chemist, you spend a lot of time in the lab. As you have different responsibilities as a project team leader or different roles on different projects, then you may spend more or less time in the lab actually synthesizing molecules. But we try and make sure that people all still spend a significant amount of time all the way up the ladder, as it were, in the lab making compounds. Other parts of my time will be sat at the computer designing compounds, having conversations with my biology counterparts, people in the pharmacokinetics groups, people in preclinical safety. I spend a bit of time making slides. I spend a bit of time coordinating meetings and sitting in meetings. And again, the ratio of the science to the administration changes depending on your role. If you're more management role, which of course as your career grows and advances, sometimes you tend towards the management side, and then you spend a lot more time dealing with that. But we try and always be engaged in the science, and so whatever level, we're still trying to do most of our time spending on the science, and at whichever level, either in the lab or in the meetings or on the computer.
So for your position, you have your background is really in synthetic chemistry. Would you say that's true of most of your colleagues, that they are also trained in synthetic chemistry? Is there anybody who's come in more on the biology side and sort of become does more synthesis now, or is everybody kind of strictly a synthetic chemist?
Yeah, sorry, I didn't specify my PhD, right? My PhD was in synthetic organic chemistry, actually heterocyclic chemistry. The vast majority of my colleagues in medicinal chemistry, not just in Novartis, but across the industry, I would say, are trained synthetic organic chemists in various angles of that, either in methodology or natural product synthesis, total synthesis, or heterocyclic synthesis. That, I would say, is probably over 95% of the medicinal chemists in industry. There's a chunk of people who have been trained in medicinal chemistry in academia as well. There's very good medicinal chemistry schools, of course. There's not very many examples of people shifting from different trainings, so they come in, like you say, from biology or from something else. No. The reverse sometimes has been the case. We actually have somebody pretty high up in Novartis who's moved from chemistry into heading up the biology group, but the reverse I'm not aware of.
Okay, great. So at what point of the drug discovery process do synthetic chemists enter the drug discovery picture? Is that during initial screening, hit filtering? When does the synthetic voice come into the discussion?
I mean, as an experienced medicinal chemist, we try and get involved as early as possible, even in some of the nascent ideas, proposing the ideas with biology colleagues or independently as teams, trying to look for opportunities, either because of literature precedents or because of internal research that's going on. What's the first time that we touch a round bottom flask and actually try and make a molecule? That's more in terms of the validation of the target. Sometimes we'll make tool molecules, they may be known from the literature, or maybe we found something from some doing exploratory chemistry, exploratory biology as well. We'll make one or two tools, and so we will still be even then involved very early on in the program. It may be two or 3 years before any real hit to lead or lead optimization chemistry is going. And then we carry on past that. We'll talk about the optimization stuff, I guess, in a little bit more detail, but past that, once we've identified the candidate and handed it off to the development organization, as it were, to try and make it, see if it makes it all the way through to a drug on the market, we would still try and keep in contact with that program all the way as far as we possibly can, at least to phase 2.
Okay, excellent. So when we talk about this particular unit is on lead optimization.
Yeah.
Out of a lead discovery program, how many leads would you ideally identify through that process? You know, there's a resource limit, but you think you'd want more than one. So how many leads would typically be advanced from the program?
Yeah, if you look in the literature, a lot of people talk about 3 to 5 as being kind of a nice number because it gives you some diversity and it gives you some scope. And like you say, if something goes pear-shaped with one of those, then you have other things that may be looking reasonable. But it's not so many that you can't possibly handle it. In reality, we can have everything from 0 hits out of a high throughput screen to 10,000. And at some point you need to figure out which of those are real and which ones aren't. But we try and pretty rapidly get down to a reasonable number. On some programs we've processed over 50 different chemical series through validation, I would say, which means in some cases you would make 30 to 50 compounds in that series before you realized it wasn't a real hit.
So what are the traits you would look for in a compound that would cause you to advance one or decide that one series would no longer be advanced?
Yeah, I mean, it's a combination of things. And actually, I don't think it's really, there's no real one-size-fits-all answer to that. It depends on what your target is and what your disease is and what your mode of delivery is. If it's an oral drug you're aiming for, then you want certain types of properties, good solubility, good permeability, good oral bioavailability, etc. If you want an IV drug, then obviously solubility is now even more important, but maybe oral bioavailability is not interesting to you, and different types of permeability are something you'll look for. Inhaled, if you look for something which is an inhaled active compound only in the lungs, then you look for actually poor oral bioavailability and something that's highly cleared systemically. So there really isn't a one-size-fits-all, but in general we tend to try and find compounds which have reasonable molecular weight, somewhere in a range of 350 to 500, somewhere around there. We try not to be boxed in with the rules and regulations because there's so many examples that don't fit those criteria. But yeah, reasonable solubility, because that helps with all of your assays and understanding of those. If you have something which is poorly soluble, and it looks clean in all of your profiling panels, for example, looking at off-target activities, it may just be that it wasn't soluble in the assays.
Excellent. So how many, when you're working on a lead, how many chemists typically might be involved in advancing that one lead or this handful of leads for a particular drug program?
Yeah. That's another it depends answer. And I mean, there's lots of reasons for that. It can depend on how high a priority the target is to how easy the chemistry is on that particular template. If you have pretty easy chemistry and things are coming together quite nicely, you may only need 3 or 4 chemists and you can actually make a lot of compounds. You may have some very complex chemistry and it's very tough, and it's a land war, and you need 20 chemists in order to make any progress at all, right? So I've definitely been on programs where there's only been 3 or 4 of us, and my last program there was 23 chemists for 2.5 years.
So
we were doing a lot of chemistry.
When you say a lot of compounds, you're talking about making compounds individually. How many is a lot of compounds by your definition?
Well, we stopped making libraries pretty much. Not these, 10 years ago, 15 years ago, people were making lots of combinatorial large libraries, 10,000 compounds. All of them were horrible, and none of them had any decent properties, even though they might have been potent on the target. We stopped doing that. People do a lot more focused libraries if you're in libraries. When we get down to the point of single compound synthesis, a lot of compounds for me is in the thousands. So any one particular drug discovery optimization program, you may make anywhere from 300 or 400 compounds to 2,000 or 3,000 compounds over the course of a couple of years in order to identify a compound which has most of what you need in order to go and test your hypothesis in people, preferably.
Okay, that's very helpful. What sort of tests are typically requested on new compounds? So as new compounds come forward, what do you request from the biology group and from different testing groups?
Yeah, that's, again, but by project you need to figure out what's the most important data, and some of that you front load. So we always set up a testing funnel, a pipeline of tests that you want to go to. Some of those you'll do immediately upon synthesis. Some of those you'll do once that first battery or tier of testing data is available, then you will go to secondary assays and maybe even tertiary assays. Around about that time is when you start looking in vivo. But the primary assays tend to be some sort of biochemical assay, a binding assay, or a biochemical functional, a biochemical binding or activity assay. Solubility, usually, some sort of high-throughput clearance assay in microsomes, and maybe a high-throughput permeability assay like PAMPA, which is an artificial membrane looking at passive permeability. So those 4 or 5 things you would tend to look at up front. Sometimes you would do a cell-based assay as your primary assay, and so that would be in your tier 1 as well. If the data that you require from those assays looks good, then you would move to your second tier. And in that, you would have maybe more involved permeability, more involved clearance data, maybe looking at things like CYP inhibition. Cytochrome P450 inhibition in the liver is a big drug-drug interaction flag. So we'd start looking at things like that, multiple different ways that you can interact with CYPs, and also the cell-based assay on your target if you haven't looked at those already. And a lot of that is really gearing up to be going into some in vivo studies, which you would get to in your like third tier, as it were.
So when you request these studies, what is the turnaround time? How long do you have to wait before you get feedback to inform you what your next compounds might be?
Oh, absolutely, yeah. Normally it's 1 to 2 weeks. Some of the data can take a little bit longer of that, but if your first tier data takes more than 2 weeks, you only get 20 cycles in a year. That's really not that many investigation optimization turnarounds. So we try and get it closer to a week if we can, and you're doubling then the number of iterative cycles per year. We don't try to be very dogmatic or very linear in the way we think, and so you're testing 50 different hypotheses simultaneously, and you don't wait for the answer from hypothesis 1 before you start testing hypothesis 2, you know. So as data is going in and you're waiting for it on one thought, you have other thoughts going along those compounds you're making, and at the same time you have compounds you're designing with a third idea, and you've ordered some intermediates that you need for the synthesis of a fourth idea. I mean, all of this stuff is all happening in concert with each other, all sort of mixed together. Sometimes it's fun trying to keep everything lined up, especially when there's 20 chemists.
I'm sure that does keep everybody busy. So when you make these compounds, about how much compound would you prepare for these typical tier 1 or tier 2 level screening?
Sure. I mean, you can get a lot done with 1 milligram. And people are surprised by that. Usually, if you make 1 to 2 milligrams, you can do your first tier and most of your second tier assays with that much material. The problem comes down to things like accurate weighing and accurately understanding the concentration of the solutions that you make from such a small amount of material. And so if you do get data that looks promising, you really need to resynthesize it on a larger scale and repeat those tests and make sure everything looks true. But assuming that that was fine, what happens usually is you start a program, early days you may make 1 or 2 milligrams because you may not have as many chemists, and also you don't really know if that series is that interesting. Once you have an established series and you have some reasonably robust data and you feel very confident, then we tend to increase the amounts that we make with our first synthesis, because it saves a lot of time in resynthesis. We don't need to wait 2 weeks to get the data and then decide we need to remake it and then reorder the starting materials, then scale up the synthesis. Some of our syntheses may take 2.5 months. You're doing 20-step synthesis for a certain analog, so that can take a lot of time to do a resynthesis. And in those cases, we tend to aim for more like 20 to 50 milligrams and sometimes even 100 milligrams. You can get a lot done with 50 milligrams of material. An in vivo PK study in a mouse only takes 10 to 15 milligrams of material. Some in vivo studies that you want to check for either a pharmacological effect on a pharmacodynamic marker, like blood pressure or something like that, or an inflammation response. If it's just a single dose assay, again, 10, 15 milligrams is going to be enough. But if you needed to do maybe 2 or 3 weeks looking for some efficacy marker, or it takes a long time to affect a pharmacodynamic marker, you need more like 200 or 300 milligrams for those in vivo studies. And if you do those in rats, they're a lot larger than mice, and it takes a lot more compound. But I mean, all the way up, once you start talking about larger species and further progressing, 50 grams of material we make in our labs in the medicinal chemistry groups, but good compounds that we need to get a lot of data on.
So at what stage would the synthesis of a promising compound transition from the medicinal chemistry group to more the process R&D group?
Yeah, and this is set up slightly different in different companies. Some companies they only have the 2 groups, you know, and ne'er the twain shall meet sort of thing. But most companies have now transitioned to a kind of an intermediate group, a scale-up group, before it ends up in chemical development. And so we tend to, in Novartis, and I know other companies I've worked at as well, once we've identified candidate compounds that we think may have a chance of being chosen to go towards the clinic, then we start talking to the chemical development group earlier than that. But once we have the molecules, we tend to make more of the 10 to 50 grams maximum. And as soon as we need more than that, it moves to either the scale-up group or chemical development at that point. Before starting clinical trials in humans, usually you need somewhere around 500 grams to a kilo of material, and we just can't do that in round bottom flasks in our labs. So they have 20-liter reactions and stuff. Again, a lot of the time it's not really the final step to make that 500 grams that's the problem. It's the fact that you have a 20-step synthesis and maybe only 70 or 80% yield at each step, and if you go all the way back to the beginning, you need to start with 18 kilos of one particular starting material. So yeah, that's when it becomes much more chemical engineering type scale.
Definitely. Well, Julian, I want to thank you for sharing your expertise. I think you gave a great short capsule of what it's like to be a medicinal chemist at a major pharmaceutical company from the start of the process to handing it off to the next stage of development. So I want to thank you, and thank you for your support for the course.
No problem. It's lovely to speak to you, Professor Allen.
Great. Thank you. Okay, that is it. I'm stopping.